Welcome back, students. In this lecture, we are going to study about the chromatography. The word chromatography originates from the Greek word chromatos, meaning the color writing. Chromatography is the laboratory technique and a separation method that is used to identify, separate, and analyze the individual components of a complex mixture. It operates on the principle of differential distribution, where the components in the mixture interact differently with two different phases, the stationary phase and a mobile phase. As the mixture is passed through this system, the components are carried at different rates, leading to their separation. Chromatography is widely employed in various scientific fields, including chemistry, biochemistry, pharmaceuticals, food industry, environmental science, and much more for tasks ranging from compound purification to quantitative analysis. As we discussed about the chromatography, now let's study about how chromatography typically works. Here are some of the key points that will help us to learn more about chromatography. The first, we have stationary phase. This is the solid or liquid material that is fixed within a column or a flat surface like a paper or thin layer. The stationary phase can be a polar like water or can be a non-polar that avoids water. Depending upon the specific separation requirements, we use them. And the next, we have mobile phase. This is a fluid, liquid or gas that moves through a stationary phase, carrying the sample mixture with it. The mobile phase can also be polar and non-polar, and it interacts differently with the components of the mixture. The next in the line, we have sample application. The mixture to be separated, called sample, is applied to the stationary phase. This can be done through various methods, such as injecting a liquid sample into chromatography column or spotting a sample onto a chromatography paper. The next in the line, we have separation process. The separation process, as a mobile phase carries a sample through a stationary phase, the different components of the mixture interacts with both phases. Some components will tend to stick more to a stationary phase and will move more slowly through a column or paper, while others will move more quickly because they interact less with the stationary phase. The next in the line, we have detection and analysis. After separation, each component of the mixture exits the chromatography system at different times. This separation can be detected and quantified using various detectors, such as ultraviolet spectrophotometer, mass spectrometer, or fluorescence detectors. The resulting data can be used to identify and quantify the components of mixture. Chromatography comes in many forms, each tailored to specific separation needs. Now let's see how many chromatography techniques are used in the industry. The first, we have gas chromatography. The gas chromatography uses a gaseous mobile phase to separate volatile compounds. And next we have liquid chromatography. The liquid chromatography, as the name suggests, uses a liquid mobile phase, often with a high pressure system, which is called high performance liquid chromatography or HPLC, to separate a wide range of compounds. The next in the line, we have thin layer chromatography, TLC. It's a simple and a quick method where the stationary phase is a thin layer of material on a flat surface, usually glass or plastic. And next we have paper chromatography. Similar to TLC, thin layer chromatography, but uses paper as a stationary phase. And the next method we have is ion exchange chromatography. Ion exchange chromatography separates ions based on their charge. And the last, we have affinity chromatography. Affinity chromatography separates molecules based on their specific binding to a ligand attached to a stationary phase. Chromatography is widely used in analytical chemistry to identify and quantify compounds of complex mixtures. in the purification of substance and in the various research and industrial applications. It plays a crucial role in the fields such as pharmaceuticals, food science, environmental monitoring and forensic science.